Okay, we'll get started. Thank you all. Sorry, Thank you all for coming. This is meant to be a very informal discussion about your graduate school options. In particular, one that we're promoting is a biomathematics PhD here at UCLA, but feel free to ask questions about any type of graduate school questions you have. So I guess I'll get started. So I'm Eric Sobel. I'm the Vice Chair for Education in the for the Department of Computational Medicine, which hosts the PhD in biomathematics. It's a historical anachronism, I guess, that the PhD program and the department have different names. The department also hosts other graduate programs, as well as the PhD in biomathematics. The other person that you'll want to, if you haven't already contacted or have the contact information for, is Samantha Andrews, who's sitting in the back. She's our student affairs officer, so if you have questions either now or later, feel free to ask either one of us. Well, that's interesting. There we go. As I said, we do want this to be informal, so feel free. We often get a lot of questions about graduate schools in general. You know, why should one go for a PhD? And we can talk about that if you wish. I certainly want to congratulate you all for being interested in computational biosciences. It's a very fast-growing field currently. Most importantly, there's many interesting, solvable, and useful problems that graduate students and faculty can work on, which is wonderful. No matter what program you end up going to, I'm sure you'll be in high demand when you're finished. So why should you consider UCLA's program? Obviously, we think we've got a world-class program here, and there's three ingredients that, to me, make up are the most important overarching ingredients for a computational bioscience program. Excellent mentors, which we have in abundance, world-class biomedical, mathematical, statistical, and computer science expertise on our faculty. And you'll be hearing, after this brief overview that I start with, brief descriptions from the faculty of some of their research projects. Another resource that you need is for a good graduate program in computational biosciences is, of course, high capacity computing resources, but of course, cloud computing makes that almost ubiquitous these days. But the third item is real data to work on. And having, let's see, so in combination with these three, and analogous maybe to these three different areas that make up a good program, there's three different resources you need to make up a good graduate career. You need to make up a good graduate research project. Let me put it that way. There's the quantitative methodology that we're all interested in, but there's also basic laboratory science, and there's also either field work or clinical work in medical fields that give you the data that you use in your projects. And having the hospital on campus that we are, many of us work with is a really outstanding resource. So why the biomathematics program amongst all the computational bioscience programs out there? There's certainly a lot of overlap in the faculty amongst the computational bioscience programs. When we introduce the faculty, that will give you their brief discussion of their research topics, you'll see how many departments they're all in. So there's tremendous overlap. And that's really to your benefit. When you have your doctoral committee, you will want to have mentors from many programs. They give you different perspectives on your research. They can help you understand the breadth of the field that's out there. So there's a lot of overlap amongst our faculty members. There's a lot of overlap in the research questions that we address in the different computational bioscience departments. There's a lot of overlap in the methods that we use, but there's also significant differences in the programs. So the Biomath program our primary goal is to train you to develop, test, and implement new mathematical models of biomedical systems. It's designed to be a hub for the collaborations that are necessary for advancement in modern biomedicine. As I said, you need not just the quantitative work, but also the basic laboratory work and also field work or clinical data. And to enhance these collaborations, we do make sure that our students are well trained in not only the quantitative sciences, but also the biomedical domain that they plan to work in. Not to the same level as the quantitative sciences, but to a significant enough level that you can communicate well and collaborate well with the experts in that domain. There's many other excellent computational bioscience programs, but they will have a different perspective. Some of them their primary interest may be in data analysis or computational considerations, but for Biomath, the mathematical model is primary. So when you, as a graduate student, in close collaboration with your mentor and your colleagues, you'll be writing original research papers. And these papers will usually stress, if you're the Biomath student, will usually stress a new analysis method, and then also include examples of how those methods are applied to real biomedical problems. In case you're wondering, the basic paradigm is that a doctoral thesis is 3 papers. They don't all have to be published, but they all have to be well on their way to being published. They have to be submittable and publishable papers. Basically 3 papers and some discussion of how they fit together is what usually makes up a dissertation. So our bottom line is the bottom paragraph here. If you enjoy sophisticated mathematics, but want to see your research quickly applied, then you should definitely consider our program. As I said, alongside the world-class mathematical, statistical, computer science expertise, our faculty are experts in a very diverse range of biomedical fields. Some of them are listed here, and you'll hear descriptions of research in these areas, in some of these areas after this overview. We are certainly a very small department, small program. We have around 30 affiliated faculty, but as I said, almost all our students have members of their doctoral committee that are outside of our faculty as well. And sometimes even the primary mentor is outside our faculty, which is fine. We do make sure that the quantitative methodology is a primary component of the dissertation. We have less than 20 total graduate students, 3 to 6 arrive per year. And I often get asked, what's the background we're looking for? What set of courses are we thinking people should have? And there really is no rules. We have some of our students come in with a strong math background, quantitative fields. Some of them come in with a very strong biology background, but are very interested in quantitative fields, and they go on to do great work. some with a mixture of both. And obviously, there's no right way to do science. Your background, there's no specific background that you need to do great science. I have here an example. The biological domain that interests you will determine which areas of mathematics would be most useful in your research. But the unifying theme is that you enjoy using sophisticated math in your application to the biomedical problems that interest you. You shouldn't feel at all inhibited to asking the programs that you're interested in, perhaps applying to, questions about their program, things like time to degree. Don't feel that you're being too inquisitive or anything. You're doing your due diligence. It's going to be a few years that you're in your program. It's an important decision for you. So do your due diligence and ask the questions of the people that you're going to be working with potentially. So our mean time to degree is slightly under 5 years over the last 20 years. Our program does include coursework in the mathematical modeling theory and application, but our requirements are really very flexible. We take into consideration the coursework you've already done and what your areas of interest are. Another point that you should ask any potential programs you're interested in about funding. So we fully fund all our PhD students, and that should be true no matter where you go. As a computational bioscience graduate student, your tuition fees and stipend should be fully covered. At UCLA this year, our stipends are $37,500 per year. It's certainly likely to increase next year. The source for this funding is certainly departmental funds, but also funds from several training grants that I run one, Bogdan runs one, who will give a short description of his research later. There's teaching apprenticeships. We only require a single quarter, and it's not really a TA-ship what we require, but certainly if you want to do a TA-ship, that will satisfy our requirement. And it's important to do at least one, if not a bit more, just to have it on your resume because wherever you go next is going to want to see that you've at least had exposure to that. Some of our students discover that their passion is for teaching, and they go on to devote a lot of effort to their teaching. But others, of course, are more interested in the research, or you can do both. Other sources of funding, certainly your primary mentor's research grant will be a primary source as you progress through your degree. Many of our students get individual NIH and NSF graduate fellowships. So that these training grants, NIH and NSF awards them to the institution, UCLA, and then we award them to students. But you can, as a grad student, apply, and many of our students receive individual level fellowships directly from NIH and NSF, and those, of course, look great on your resume. I mentioned your CV, just to let you know, in academia, that's what a resume is called, your curriculum vitae, your CV. Let's see, we have many seminar series to expose you to the breadth of projects that are available. We are, this year, we will have a seminar series run by and exclusive to students so that they can get honest, constructive feedback in a low pressure environment. We have courses in career development to train students in the non-science aspects of a successful career in science. And we also have collaborations with several companies, commercial entities that provide a terrific access to the types of real-world problems that they work on, and exposure to what that kind of work is like. So our coursework, there's deterministic modeling, stochastic modeling, systems biology, statistical data analysis. And obviously you don't have to take all these, but these are some of the core courses that we certainly encourage you to take. And then there's, that's sort of the theory of modeling. And then we have applied modeling courses in statistical genetics, mathematical oncology, and many other areas. So where do our graduates go? About 50% of them are currently either academic or medical faculty position. They hold these positions. And there's another 15% which are still in postdoc positions. And so they may or may not end up in a academic or medical faculty. Usually their own choice once they've decided what they want to do. Usually they go into a department, obviously that not many biomath departments around the country or the world. So they usually end up in a department of their biological domain of interest. For example, I'm in human genetics as well as computational medicine, but their research is still mathematical modeling because every biological area these days know that to make advancements, they need a mathematical modeling as part of their tool set. The roughly half that are not in academic positions are still in computational healthcare fields. Just a couple examples. One of our graduates is works in the brain genomics division, Google Research. Another one is a co-founder of a company that does AI in healthcare. So that was my overview. Certainly happy to take any questions if anybody has any, including out there in the Zoom world. If not, I will. Oh, I'm sorry. Thank you. Yes, please. I'm sorry. Say it again.
Do you have to take any lab course or like chat training in this program?
So the question was, do we require any laboratory courses? Not laboratory courses. No, we do require you to take some biology courses that get you up to speed in the domain of your interest, but not laboratory per se. No. Yes, please.
Hello, my name is Lucas. There's some skills.
I'm sorry, just 1 second, Lucas. I'll repeat the question. There's a question in the audience here first. I'll get right back to you. Sorry, say it again.
Yes.
There's a lot of overlap in the, as in, I think just about every one of the faculty members that you'll hear give a brief description of some of their research is also in bioinformatics. Our researchers tend to be the more theoretical side of bioinformatics, the people who are also in computational medicine as well as bioinformatics. So we're more on the theory side. We more stress the modeling as opposed to maybe a bit more on the computational algorithmic side in the bioinformatics world. Bogdan, do you want to have any other comments on that along this?
I think the easy way to distinguish it is if you're interested in developing methodologies, computational methodologies, then Biomath is the way to go. If you're interested in applying them, some real biological data, then that's more on Biomath.
So Bogdan very succinctly, eloquently said that if you're interested in, just repeating it for the Zoom audience, if you want to design quantitative methods, wasn't quite the words you used, but I think that was the gist, then Biomath is more appropriate, or is appropriate. I'm sure you can do it also in bioinformatics if you really wanted to. But if you're more on the applied side, you want to apply the methodology, then the bioinformatics is certainly an appropriate field. Thank you. Lucas, what was you mentioned, sorry, I cut you off in the Zoom ether.
Oh, don't worry. I didn't know there's some people there with you. There are some skills do you think the people who pretend to apply to mathematics have to do, like math labs, programming with Python, or just math skills?
You can certainly get a biomathematics degree doing minimal computing if you wanted to. You can, some of our graduate students, their primary mentors are professors in the math department. Some of them are also in our department as well. But the point is that you can be doing extremely theoretical work if you desire it. There's a very broad section of research options in our department. Most people, I must say, most of our grad students have, they build a mathematical model, but then they apply it to actual data, which means some degree of implementation in a computing environment, but they don't all do that. There was a question in the chat. Sorry, I can't seem to see the chat in my view of the world here. Here it is. Do biomath students undergo lab rotations? Oh, that was, well, okay. By lab rotations, I mean, do they, a lot of graduate programs for the first year, each quarter, they're in a different group. And so they get exposure to the work in that group. The answer is no, we don't do that. We don't have formal lab rotations or group rotations, since we don't really have labs, most of us. We encourage you from day one to get involved in research. Some of our students come in knowing what area they want to go into, and they get heavily involved from almost day one. But most of our students are looking around and seeing where there's a good fit, both scientifically and culturally, or in which group, both the biological interest and the math interests. And so we encourage you to, as I said, we have a lot of seminar series to give you exposure to different projects. Of course, you talk to your biomath colleagues, and you do take courses from our instructors, from our faculty, and you see what kind of things they're interested in that way. You can also start going to lab meetings and seeing what projects people are working on. And if you see that you have some interest, you can ask them to let you start doing some either background reading, guided background reading, or actually on some small projects. So, but the answer is no, we do not have formal lab or group rotations. Thank you for that question. So I'm going to hand it over to Jamie Lloyd Smith, our first faculty speaker. Dr. Lloyd Smith is in EEB, Ecology and Evolutionary Biology, as well as Computational Medicine. So I will. Is the PDF okay?
Whenever. And given the numbers, sort of in 5 minutes is good.
No, anything like that is good. 5 is fine.
Great. Okay, hi everybody in Zoom, in the real world, etc. Amy Lloyd Smith. I'm in ecology and evolutionary biology and computational medicine. My lab works on infectious diseases, and most importantly, how new infectious diseases emerge. So I used to have to give a spiel on why this is important, but we've all been living this. As we all now know, lots of new human diseases enter the human population from other animal species. There's a lot of really interesting biology that is involved in that. And my lab tries to apply a range of quantitative tools, mathematical modeling, statistical techniques, really whatever we can throw at it, to assemble ideas and mechanisms across scales from like molecular processes right on up to population ecosystem type processes to think about how this process of pathogen emergence happens. So there in the top corner there, this is like when I started in this was SARS-1, coming out of a civet, getting into humans, causing the beginnings of a pandemic that we're able to stamp out. Now we've all experienced SARS-2, which caused a full-on pandemic that continues to torture us. In my group, we go way back to the original SARS. We've been caught up in the current one, as is the rest of the world. We also do projects that I've kind of binned here into 3 broad classes, but it's a little bit more blurry than that. A good chunk of the group works on what you might call classical disease ecology. So looking at how pathogens circulate in wildlife populations. And we have a well-established long-term study of this bacterial pathogen, Leptospira, circulating in a handful of mammalian hosts in the California coastal ecosystem. So here you see, these are California sea lions. This is a time series spanning several decades of outbreaks in California sea lions. This is a California Channel Island fox and an island skunk. That's a little outbreak of the same pathogen that broke out on one of the beautiful islands off the coast here. So we have people in the group who actually chase these animals around and catch them and get samples. I don't have a wet lab, but we have lots of friends who do. So we turn those samples into data, and then we do a lot of modeling and statistics to try and figure out what's going on. And this is expanded now to the wildlife species all around us in LA, who also have this pathogen, and we're trying to stitch it all together. Everything from population level dynamics to genomic data to thinking about how antibody titers rise and fall in animals and so on. The next little bin I've got here is transmission dynamics of the animal-human interface, and I'd really broaden that to any cross-species transmission because it all follows similar rules. I've been thinking about this and working on it for 15, 20 years now, which involves everything from kind of developing theoretical frameworks for what are the, how do the stars need to align for a pathogen to get out of a bat or a pig or whatever into a human and possibly initiate an outbreak among humans. And then we've worked on various case studies, both between animal species and things like how do people hunting wild mammals for protein in Central Africa, how does that give rise to spillover events of viruses, monkeypox, in its original sylvatic reservoir. And then the third category, which is something I'm increasingly into, and we have someone in the room who's contributing to this effort as a fifth-year master's student, is thinking about what I call the within host ecology of really viruses viewing, the inside of a single host individual as a landscape. And so an infection event is really an ecological invasion of this virus across this heterogeneous landscape. And you can apply a bunch of ideas from theoretical ecology and evolution to what's going on inside individual host. So we're thinking about all of that in a sort of spatial way, bringing in insights from physiology and molecular biology that inform the fitness of the virus in different body compartments, and also trying to kind of dissect a single transmission event and think about all the things that need to go right for one host to infect another. A bunch of wonderful people in the lab are working on it. Am I out of time yet on my first slide? Quickly, just some of the modeling frameworks that come to bear. This is a cartoonish version of the famous SIR model, where you divide a population into people who are susceptible, people who are infected, and people who are recovered. And just by taking this diagram here, translating it into differential equations and turning the crank, you can get a situation where if you introduce an infected person into an otherwise susceptible population, this little model will give you a nice little outbreak, little epidemic curve, which goes up, comes down, tails off. And of course, this is the most basic form of this population dynamic of epidemics, but we then elaborate on it in ways that correspond with the biology we're trying to represent. And we use that for a range of different things. I won't say all these. I'll emphasize that we don't just use this to predict the future. That's one thing that gets a lot of headlines, but mostly we use it to try and understand the mechanisms that give rise to patterns that we see. And then I would emphasize down here, estimating key parameters. We build models that represent the mechanisms we think are important as a way of doing the best inference we can, like getting the best value for your buck from data to learn about the actual processes that are driving these large level phenomena like epidemics or emergence events that we care about. I'll also highlight this one, linking data across scales. We build mechanistic models that try and connect what's happening in cells to what's happening in tissues, to individuals, to populations, and so on. It gives you a formal way of doing what you're otherwise stuck sort of trying to wrestle with your intuition. A little picture of that. This is just zooming in on this diagram of all the stuff that goes on for a transmission event to happen. So this person has got some viruses propagating in her airway. You know, you can think of that in terms of the spatial structure of the lungs and the upper respiratory tract and so on, or you can break it down to the actual sort of networks of cells or the molecular interactions underlying that. All of this, you can kind of propagate the dynamics up and think about how that gives rise to viral shedding into the air or into whatever other medium is important for transmission of that pathogen. That virus will then, you know, it needs to be out there in the world for a little while. It might decay, it might inactivate in its time between hosts. And then this fellow comes along and gets some kind of inoculum. And then you have to think about how does that little invasion process play out in the new host. And so by really trying to deconstruct this transmission event, it gives us a way of linking all the work that's going on in sort of wet lab, virology, biomedical labs, understanding what these viruses are doing and, you know, how this variant's different from that variant, and trying to link it up to the epidemiological outcomes of, oh no, we've got the hypertransmissible Delta variant, we've got the immune-evasive Omicron variant, all that stuff. So we're trying to sort of construct a mechanistic framework that does all that. Final slide is just a handful of examples. I'm not going to dwell on them all, you can read them. But the types of things that my lab has been involved in, up here you've got, this is 2 different flavors of avian influenza. It's all kind of alphabet soup depending on their surface proteins. But there were some curious age patterns, and we figured out that that was actually driven not by age, but by the year that people were born and what their early life exposures were to seasonal influenza. And that ended up, we're able to establish evidence of a type of immunological imprinting, which has repercussions for your exposure to future emerging flus. Over here, you got some cross-scale evolution, thinking about what's going on inside hosts and how selective pressures within a host either do or don't propagate across populations, depending on how many and what mix of virions transmit from one host to another. Down here is a COVID project looking at how long the different how long the virus is able to remain viable in different environmental conditions, different temperatures and humidities. For the temperature effect, this is empirical data from some collaborators. The temperature effect made sense. The hotter it is, the quicker it inactivates. The humidity pattern was a bit weird. It's nonlinear and U-shaped, as you can see. So we ended up creating a sort of a biophysical chemistry model of what's going on inside droplets as they evaporate and how that may be affecting whatever chemical reactions drive an activation. That turned out to fit the data and predict data out of sample. And this is a big time series analysis of what's going on in sea lions, kind of a clumsy plot, but you can see in red, that's observed data of how intense outbreaks of this Leptospira pathogen are year to year. Then we fit a series of different models to understand what factors drive the intensity of those outbreaks in the sea lions. And the best fit we get calls on both information on kind of how many susceptible sea lions have been born lately, and also what are the environmental conditions that may mediate the contact and transmission process between them. And that is the blue, the dark blue plot, which you can probably not see. And that gives you a flavor of the kinds of things we do. I would be happy to take questions, but I think we should rather turn over. And then I have to apologize that I have to leave before this session ends to pick up my kids. But there it is, pandemics.
Any last one quick question, or we'll just move on? Thank you, Jamie. Bogdan, you're next. We're basically doing an alphabetical order. What's this one? Bogdan is Bogdan, Dr. Postdata is a professor in computational medicine, human genetics, pathology, and bioinformatics. So as we will you'll see that everyone is heavily cross work.
Great. Thank you, Eric. I guess the first thing to learn about UCLA is that we go by first name. So I'm Bogdan. It's good enough that my name is kind of unique, and it's going to take you guys a long time to learn it. But once you learn it, there's no other Bogdans at UCLA. So, that identifies me as of now. Hopefully they'll be in the future. Okay, so I'll show you a little bit of the flavor of what we're doing in my group, and by extension of the types of research that you'll be doing as a biomathematics or more generally computational medicine PhD student here at UCLA. And so my lab is mostly interested in genomics or computational methods for genomics. And kind of as a snapshot of all the types of groups that you'll find here at UCLA and beyond, is that you'll be embedded in a bigger community that's interested in various different topics. And just to answer more directly the question that was raised before, your job as kind of a biomathematics or computational medicine trainee would be to develop methodologies and be at the forefront of how can we develop methods anywhere from helping other people in the biomedical space. So here it's listed a bunch of people that were in my lab at the time, maybe 6 months to a year ago, and people that have graduated. And you'll see that each of them has their own training program or their own expertises, anywhere from bioinformatics to genetics and genomics to, let's say, other types of programs that are more applied. So you will be trained as how to develop mathematical models, how to develop computational solutions to problems that are in the medical domain or biomedical domain or biology as a whole. So my lab features 3 research themes, and I'll apologize in advance that I won't have a lot of time to go into each of them, but hopefully Samantha or Eric can give you guys access to these slides so you can go through them in more details and shoot emails or ask people in the audience about them. So we're very much interested in computational methods for functional genomics, for underrepresented populations, and nowadays, for the past 4 or 5 years, we got very interested in medical records. And I'll show you what I think is the next generation of genomics, where we have data, genomic data from our patient populations here at UCLA, and how do we essentially ask the questions directly with patient, with patient translation or patient application. So functional genomics, there's a huge room for methodology in this space. On our domain, we were kind of inventing this technique called transcriptome-wide association studies that essentially relies on a simple computational technique of predicting gene expression or the gene amounts of products of mRNA based on the genetics of a particular individual, and using that to essentially come up with new data through this procedure called imputation at scale. And then once we do that, then we can correlate this predicted gene expression with disease outcomes. And that gives us a direct way to identify genes that might be risk genes for various different diseases. And I'll link you, you can read more about this type of technique in this citations. And in this place, going forward, we're very much interested in how do we take this knowledge from complex gene regulation all the way from isoforms to splicing, all sorts of different, gene regulation is very complex. How do we model that mathematically, and how do we bring it back to, disease outcomes through these techniques called genome-wide associations scans? A different area here that we're very much interested is in single-cell transcriptomics, and how do we take single-cell transcriptomics and take, ideas that we are familiar with from polygenic risk scores and large-scale genomic studies and project them back on a single-cell basis. So essentially there, can we say that a particular single cell is the causal cell for a given disease? And finally, there's a whole range of projects in my group where we're interested in mathematical modeling of figuring out how do we reduce the cost to generate more data at scale. And you can read more about it in this works that I'm highlighting here, or you go to our website. The other part that it's kind of fascinates us for the past 10, 20 years is this idea that we need to do a better job of computational methods as well as data for individuals that have genetic ancestries that are admixed. And this slide is just simply to show you that
when we think about individuals of different genetic ancestries, we typically label them in different, continental groups or different types of mixture across multiple continental groups. Here I'm showing you, the impact of the transatlantic slave trade, creating populations that are labeled as, Hispanic, Latino, or African American. And underlying that, there's a growing need to, do more with genomic studies in such populations. But one of the areas that it's, in my opinion, is greatly undervalued is that we need better mathematical and statistical methodologies in this space. So, hopefully this is where you come into play and start thinking about how do we model such a mixture? How do we model all these issues that we have with potential impact for all of our patient populations? And in this space, we're very interested in how genetic ancestries interplays with social determinants of health. Probably all of you are familiar with the fact that genetic ancestry correlates with, social determinants of health, and, teasing these apart has massive implications for, how do we understand genetic basis of disease. We're very interested in how do we predict outcomes based on one's genome in individuals that have mixed ancestries. And like I said before, we're very interested in this, essentially modeling, the future, if you will, of all of us, which is that all of us will have some amount of mixture in our genomes from various different sources. And finally, I'll just present a little bit about, how we transition from current genomic studies that essentially look at one disease and collect data for it towards biobank type studies, in which here at UCLA and many other medical institutions, we started genotyping all the patients that come through the doors in our hospitals. So here it's called Atlas. So this is a biobank that it's ever growing within the medical system. It's slated to have 150,000 patient records that are linked with their medical records. And I'll show you some slides of how the first wave of data looks like. So here, as you might expect, this is a map of LA color-coded by some website based on self-reported race ethnicity. And the point here on the left side is that there's lots of colors there. So LA, as you might expect, is very diverse. It's one of the few places worldwide that does not have a majority of based on self-report. So it's a minority-driven different city. And on the right side, you see results from the first wave of genetic studies within our UCLA patient population. There, every dot is one individual, and this is a computational technique that takes one's genomes, or in this case, it's 40,000 people's genome, and project them on a 2 dimensions. So it does a dimensional reduction and principal component space, and you can already see these patterns that emerge that kind of resemble, if you will, all these different color scheme that you will see on the geographic map. This is not just what we observe here. This is also mirroring kind of what we observe in New York there on the right side. So there's an ever-growing need for methodologies to treat this diversity in the genetic ancestry space across the continuum. And in this space, we're working heavily on taking and modeling the genetic data, linking it with electronic medical records. And I haven't had time to show you, but we're very interested in how do we extract phenotypes? How do we deal with the practical implementation of polygenic risk scores? So I believe that there's a strong need for mathematical modeling on how do we actually do genetic risk prediction in the clinic in an equitable fashion. I think that there's a strong need for you guys, for all of us, to start thinking about the mathematical or the computational methods in that space, and ultimately to identify patients at high risk. I think that's my last slide. I'll stop here. And I think I'll take questions either now or after. It depends on what Eric allows me.
Sure. Well, there'll be snacks afterwards. So if you have questions
over snacks.
Yeah, please hold them for. Thank you, Bogdan. I will eventually get that into my head. Harold is next. Dr. Pimentel is in computational medicine and bioinformatics. I forget. Are there other programs? Human genetics. I knew that. Human genetics, of course. Sorry.
All right. And I guess also recently computer science as of like a few days ago. Okay, so I'm going to try to keep it short and kind of give you a flavor of the types of problems that we think about in my group. In particular, we do think a lot about functional genomics, but maybe in a slightly different space that Bogdan presented here. We think a lot about molecular regulation and like what genes are talking to what other genes. And because of that, we work very, very closely to experiments. And so I think that like over the past maybe decade, even call it 2 decades, a lot of bioinformatics has solved what I call this sort of post-data problem, where you have something that comes off of a DNA sequencer, which is really just a proxy for a molecular phenotype, and you turn it into something, right? You turn it into a plot. But it turns out, unsurprisingly, right, that there's a lot of choices that you make before you even sequence the data, right? So there's a lot of choices about maybe how many mice you're going to use, what CRISPR guides you're going to use to cause perturbations, how many cells you might extract. A lot of these are going to be random, right? Because they're going to depend on the specific mouse. And that mouse has a lot of things that might vary in its environment and these sorts of things. So we actually focus a lot on integrating this sort of computational the computational methods at every single point. So we've been working a lot on pre-data modeling, which is really trying to do like an informed experimental design. We're trying to take a lot of these like power analyses much further by thinking a lot about all the different inputs that are going into the experiment. And then we're using that knowledge to develop statistical models and computational models of what was actually represented once the data arrives, right? And so I think this sort of methodology leads to some really cool methods and really, really extracting the most out of your data. And one thing that we've been thinking about more recently is how do you adapt these models? That is, when new data arrives, how do you sort of update your knowledge so that you can make new choices about new experimental designs and sort of nominate new series of experimental designs? And this is coming from some collaborations here that we have going on with some folks in human genetics that are basically designing assays where you can do a bunch of genetic perturbations on a plate, let them run, over the week, and then nominate a new series of genetic perturbations that you might want to do and have a robot basically go and do all these things. Okay. And so what do I think about all day long? Well, I kind of like to pretend that the genes regulate each other through these really complex networks, right? It turns out that this is a reasonable model for a lot of molecular genomics. And if you believe that, there's some sort of phenotype that's at the end, at sort of the end of this mechanism, then you can sort of model this under some assumptions, right? And a lot of what we spend our time doing is really, assuming that this is the model that exists and doing a bunch of molecular experiments, obviously our collaborators doing those, and then trying to infer these edges. And it turns out, unsurprisingly, that like under all these different cell types, under different diseases, under different genetics, these networks look slightly different, right? And we're really interested in trying to understand what the similarities are and what the differences are. And so I think, hopefully there it goes. So I'm not going to spend, I was going to talk a little bit more about some of these projects, but I'll just give you the really quick overview. So one of the sort of successes that we have had recently, by the way, my lab's not that old, it's about 2 years old, so we don't have a wall of papers like Bogdan does. But one of the things that we're almost done with here is sort of a pre-data modeling of this FACS experiments. And the idea here is that you basically can knock out every single gene in the genome in a bunch of really, really parallel experiments that all happens on one plate, and then read a phenotype. In this case, the phenotype is gene expression. And it turns out that one of the things that experimentalists have been doing sort of religiously, I would say, is doing what's called low multiplicity of infection. And we don't really have time to talk about what that means, but we showed through a number of simulations that it turns out that you probably don't need to do low multiplicity of infection. And you can actually use significantly fewer material by actually, modulating a number of these parameters. And I don't have the experimental data here to show it, but you could trust me, the paper will be out soon. And because of this, we can develop some pretty cool models that that allow us to basically share information across all these different dimensions and do better inferences on the data. So one other thing that we've been thinking about a lot, right? I showed you a network. We've been thinking about sort of what I would call like core machine learning algorithms to basically assume there's some network and you have some way of doing interventions on the network. Well, each intervention tells you something different about the network. And when you sort of glue all these together in this big mathematical model, you can then start to stitch together what that network looks like. And this is motivated by real data in single cell experiments that we have a number of collaborations with. It turns out it works. Okay, and the last sort of thing, which is actually starting, we're dipping a little bit now into some of these like neuroscience type of experiments, where basically you have a bunch of, you could think of everything as a big network, and you have a number of phenotypes. Maybe that phenotype is like behavior of some population of neurons, or behavior of how you might eat or something like this, right? And so imagine there's a bunch of regulators of this behavior. What happens when you perturb those? Well, you end up learning some part of the network, and you have some unknown part of the network. And then you have to nominate another set of experiments. Well, you could do this through this sort of framework. And so we've been developing a lot of statistics on how to do this sort of thing. And with that said, we have a number of collaborations, some across a number of different technologies. Some are in like this imaging space with mapping enhancers in prostate cancer. We're also doing a number of stuff around CRISPR design, as well as in vivo experiments, and also have another project here with Catherine Plath on blastocyst development and trying to understand the genetic regulation of that. And I think that's all I have. Yeah, and just say thanks, and this is my group and some of the people we work with.
Thank you. I believe our next speaker is on Zoom. Is that right?
Yes, I'm here.
Good. And give me 1 second, Mary. Our next speaker is oh, too much. That's the worst.
Can you hear me? Okay.
I muted myself. Now you probably can hear me.
Yes, I can hear you.
I will.
But you
mute
could hear me
myself.
when you were muted?
Yes.
Okay, great. I'll share my screen. Okay, can you see my screen? Okay, can people see and hear?
Yes. Let me
Okay.
introduce you, Mary.
Okay,
Dr. Steele is in computational medicine. She's also a clinician in hematology oncology, spending maybe half her time seeing patients, roughly, something like that, and half her time doing research. So it's all you, Mary.
okay, great. Thank you. Thank you so much for having me. And I'll share just a couple of projects that we're working on. The general goal of some of the projects that I work on is to use probability and statistics to better understand the growth dynamics and risk factors for cancer progression, and to optimize treatment and prevention strategies. So we look at applied probability models from applied probability branching processes, continuous time Markov chains, and we can model cell population dynamics The reason why stochastic models are useful to us are because of the events that are of interest are rare events like mutation, either oncogene activation or the loss of two tumor suppressor genes, or development of a resistance to mutations in tissues that are exposed to treatments. We're also interested ultimately in eradicating the tumor cell population, so extinction is an event of interest. And so we can work on problems that are predicting optimal duration of treatment, which is looking at average time to extinction. Alternatively, you might be really interested in toxicities of a treatment and predict a safety of a treatment. So that's the average number of, at the time of eradication, the random time of eradication of the cancer cell population, you're looking at the number of healthy cells that are available. A couple of graduate students that are now alums, Alfonso Londeros and Tim Stutz, worked with Janet Sinsheimer, Ken Lang, and me on developing user-friendly stochastic simulation software, both in the well-mixed setting where we're looking at stochastic simulation of populations, and in the where we can, in the lattice simulator, where we can model spatial effects. So that's important. This is an example of tumor immune cell predator-prey dynamics. And then finally, another project that I'm working on involves a collaboration I have with the Komen Tissue Bank, where we look at healthy breast tissue, but we're looking at accelerations in epigenetic DNA methylation-based estimates of age that are associated with risk of carcinogenesis, risk of cancer development. We're also looking at accelerated epigenetic aging in blood of women who have early stage breast cancer and have undergone toxic treatments like radiation and chemotherapy that age the patient faster than without those treatments. And then finally, we look in lymphocytes of persons living with HIV infection. And I think that's it. So I'll let the next speaker go.
Thank you, Mary. I know. Okay, I think we're back. Our last speaker today is Daniel. Dr. Tward is, I don't think Mark is here, right, Samantha? Okay. Yes, Dr. Tward is of course, faculty in computational medicine. He's also in neurology and bioinformatics. Sorry.
Great. Thank you for the introduction. Is this microphone picking me up? I don't know how to get feedback one way or the other. Okay, that's great. I only have one slide for you, so I'm not going to keep you from your snacks for too much longer. But I wanted to talk a little bit about the major thrust of my work, which is to study imaging data of the brain to understand how the brain changes during disease and what sorts of variability is typical in people or model organisms that don't have a disease. So the data that I'm showing here today is centered around one important problem today, which is diagnosis of Alzheimer's disease. And Alzheimer's disease affects people as they get older, and as the population of America and the world is aging, as healthcare gets bigger, more and more people get Alzheimer's disease. One big problem with studying Alzheimer's disease is that you can't actually be diagnosed with the disease until after you're dead. So when people are still alive, they can be diagnosed with something called probable Alzheimer's disease, or probable Alzheimer's disease with increased evidence due to something to the presence of one specific biomarker, but we don't know for sure that they actually have the disease until after they're already dead. About 80% of people who die with suspected Alzheimer's disease, it turns out they actually have the disease. And about 20%, it turns out they had some other form of dementia, whether it was strokes or Lewy bodies or something else that has similar symptoms. So some of the work that I'm doing is looking to diagnose Alzheimer's disease as accurately as possible, as early as possible, using imaging data, which can be collected non-invasively. So on the left-hand side here, you can see some brain MRI scans of a subject starting at age 70 all the way up to age 74. This person came in for a new brain scan every year. And if you look very carefully at some of the anatomy, you notice really two key features that are changing over time. One is this structure in the middle called the lateral ventricles is growing very slightly over time. And generally what that means is the brain tissue is shrinking and the ventricles are starting to fill with fluid. So this person has neurodegeneration. But this type of signal isn't characteristic of Alzheimer's disease. There's a lot of diseases that will cause the ventricles to expand. So another change that we see is this area with a red square called the hippocampus and entorhinal cortex. And this area changes in a very stereotyped pattern, which is characteristic of Alzheimer's disease. If we wait until this subject has died and we take them to autopsy, you can see what this tissue looks like at very high resolution and zoom into the level of single cells, which you see on the bottom right. And people with Alzheimer's disease have this specific type of pathology called a tau tangle. So the goal of some of my work is to quantify the distribution of tau tangles in people who have already died and follow them back in time using information from electronic health records or larger experimental studies and try to understand how the shape of their brain in this region in particular, the medial temporal lobe, where the hippocampus is located, try to understand how that shape changes over time in people who actually have Alzheimer's disease, as confirmed at autopsy, as compared to people who have other forms of dementia or people who are otherwise healthy, other than the fact that they are aging. So to do this, we use a few different mathematical tools. We use differential geometry to study how shape changes over time. We use computer vision systems to detect and quantify the distribution of pathology over time. And we use statistical models to test where in the brain anatomical structures are changing and where those changes are linked to the presence of a disease or not. So I'm going to stop there. We have a lot of work related to these projects, both in the space of Alzheimer's disease, as well as in the space of studying the normal brain. So thanks for your attention.
That concludes our presentations. There's snacks out there, and hopefully you found there's some information in this hour, and we are all available to answer further questions. And if you can't think of them right now, please feel free to email Samantha or myself, and we will try to get back to you. Thank you. We'll conclude. Thank you all out in Zoom land. Bye-bye.